Human Genome Heredity, Module 1, Lecture 1 in the Genetics series. The objectives of this lecture is to understand the structure of the human chromosome, describe cell division, including meiosis and mitosis, recognize the difference between meiosis 1 and mitosis and the purpose of synapsis, recognize the basic DNA structure, including the double helix, replication, and transcription, and demonstrate genetic risk using Hardy-Weinberg. Human chromosomes were first discovered in 1959, and humans have 46 chromosomes in 23 pairs. Here is a picture of a karyotype. This is the chromosomes put in size order. The largest human chromosome is 1. The smallest is actually 21. But when they were doing the unbanded karyotype, they thought 22 was actually larger than 21, and they had already called trisomy 21 Down syndrome, which we'll talk about later, so they never actually changed it. The size of the chromosome, however, does not tell us how much genetic material, so the genes are on these chromosomes. The chromosome with the most genetic material is 1, followed by 2, followed by 3, followed by, that's right, chromosome 19. So some chromosomes, and the chromosomes that are the most gene poor, are 21, 13, and 18. And we will talk later when we get into more cytogenetics why that's important. Here is a male karyotype. The first one we showed was a female, and there's an X and a Y. So the Y chromosome is much smaller than the X, and there's only less genetic material. Here we have, so inside all the cells in our body, we have a nucleus. Inside the nucleus are the chromosomes. And the chromosomes are really like a scaffold that holds this genetic information, very tightly wound. The DNA, which is used as a template to make RNA, which is then used as a template to make protein. So if you think about a chromosome like an encyclopedia, or I like to think of it like a recipe book, think of a gene like a page. And when you're reading a recipe or you're reading a sentence, if the sentence is supposed to say the car was red, what if we changed in the sense of a missense mutation, the car was hat, we change a word. Or the car was, we just change a letter, red. Both of these change the meaning of this particular sentence. But if the next sentence was, and I drove to the store, the car was hat and I drove to the store, may sound strange, but still makes you get the gist of you have a car and you're driving somewhere. As opposed to in the nonsense mutation, the car was red. When there's a change in a letter, it causes a premature stop codon that causes the sentence to stop and all future sentences to stop. So instead of the car was red and I went to the store, it just says the car and that's it. And we have no more information. Insertions are, mutations are an insertion of a word or letter, and a deletion is a deletion of a word or letter. Sometimes this also results in a nonsense mutation, but can change the whole sequence of the DNA. DNA structure consists of deoxyribonucleic acid. There are 4 bases. Purines are adenine and guanine, and pyrimidines are thymine and cytosine. Adenine and thymine have double bonds, whereas guanine and cytosine have a triple bond. Gene-rich areas of the DNA have more GC-rich or triple bonds. This holds together tighter. The DNA, there are 2 strands and they're held together in the double helix. And it's arranged 5' to 3' and 3' to 5'. And these 2 strands together are known as the chromosome. The DNA is further packaged into around histone proteins. And there's about 140 base pairs of DNA wound around each histone. And each complex of DNA with around 140 times on the histone is known as the nucleosome. So there's a great deal of contraction of the genetic material in order to shove it into the nucleus. Next, we're going to talk about cell division. Mitosis is a somatic cell division. This is how the body grows. A single cell divides into two identical cells. Meiosis occurs only in gametes. There are two divisions. It results from going from diploid or 2N to haploid or 1N. There's also a special stage in meiosis 1 known as synapsis that allows crossing over of the DNA between the maternal and paternal copies that causes increased mixture of the DNA. So here we have mitosis. Normally the DNA is very stretched out when it's not wound around those nucleosomes. And this is the time that it does all the business, the work, the making the proteins. And then we're going to start division. So the chromosomes start to contract. And we have these centrosomes. They're like polar bodies that go to each side. Then they contract to the point that you can start to see them in prophase. By the end of prophase, in prometaphase, you start seeing them line up on the spindle. In metaphase, they're all lined up on the spindle. And then in anaphase, the chromatids separate. And then we have two identical cells. S phase is the replication phase. This is when DNA is replicated. So that's so then we have two copies of all of our genetic information. In replication, the double helix separates and new bases come in. This results in two identical chromosomes. The enzyme used is RNA polymerase. G2, this is actually an incredibly important stage between replication of DNA and cell division. This allows, when there's mistakes, for there to be a double check that we don't make these mistakes. And this is somatic problems. And we're going to talk about this more when we get to cancer, how this is, when this does not go right, can result in the cell making a mistake that can cause a lot of trouble. Now we're going to watch a small video on mitosis, or we're going to try.
During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2 or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and 2 centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, the structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands or spindle fibers are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the 2 ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of 2 cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the 2 halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the 2 new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The 2 new cells are now ready to grow and perform their specialized functions. During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei.
Gregor Mendel was the first botanist who did research on pea plants, and he's actually known as the father of genetics. He proposed 3 laws. The first law is during reproduction of the gametes, 2 factors segregate so that the offspring inherit one from each parent. And I would like to say this is long before we knew what chromosomes were. The second law of chance determines which of these traits is inherited from a parent to the offspring. And third, dominant inheritance is when one of the traits dominates over the other. With recessive, both are equal. Mendel's first law, the two members of the gene pair or alleles segregate separately from each other to form gametes. Half of the gametes carry one allele and the other half carry the other allele. Second law is the principle of independent assortment. The distribution of one allele into the gamete does not affect the other. So if we look at big A, big B, little b, this parent could donate big A and little b, or big A and big B, big A, little a, big B, or little a, little b. And this is random. So with two different alleles, there are all these different possibilities. Next, we're going to talk about meiosis. The purpose of meiosis is to reduce the number of chromosomes from diploid to haploid. And this is essential. And this is how we don't increase the number of our chromosomes every single generation. So you have 46 chromosomes, you donate 23 to your offspring, and your spouse also donates 23 to go back to 46. Segregation of the alleles at either meiosis 1 or meiosis 2, according to Mendel's first law. Shuffling of genetic material by random assortment of homologs in accordance to Mendel's second law, and additional crossing over done, or additional shuffling done by something called crossing over. So the interesting thing about meiosis is this division only occurs in the gametes, and there's actually two steps. So in meiosis, you have your diploid, and you duplicate your DNA, you divide it in half, and you get two cells. But in meiosis, you have actually two divisions, and you go from diploid to haploid. You end up with 4 cells. The first stage is prophase 1 in meiosis 1. And meiosis 1 is really the important one. Meiosis 2 is really mitosis, but without any cell division in between. So in leptotene, the chromosomes become visible, and the two sister chromatids start to align. So here we have one sister chromatid, and here's the other. This is dad's copy. Let's call this mom's copy. Zygotene, homologous chromosomes align, and we have the synaptolemmal complex. And this is when there's additional crossing over between the maternal and paternal copies of the gene. Pachytene is when they're tightly coiled, and then diploidine is when this process breaks down. Diakinesis is when the chromosomes reach their maximum condensation. So as I said, synapsis only occurs during meiosis 1, and it allows increased recombination between the maternal and the paternal copies of those chromosomes. So again, prophase 1 is during zygotene is when synapsis occurs, metaphase, anaphase, telophase. Meiosis 2 is really just very similar to mitosis. It's just that there's no cell division in between. And at the end, you have 2 cells that are both going through meiosis 2, and then they result in 4 offspring. So here's another picture of crossing over, big A, and this individual has big a, little b. And so they are crossing over during synapsis. So instead of there only being big A, big B, or a, little b, there could also be big A, little b, or a, big B. The further these alleles are from each other on the chromosome, or not alleles, these are from each other on the chromosome, the more likely these possibilities would occur. And this has to do with linkage. The closer these are together, the less likely you're going to have synapsis. I mean, you're less likely you're going to end up with something like that. Spermatogenesis begins during puberty. It initiates in the seminiferous tubules, and these tubules are lined with spermatogonia. The process of meiosis takes 64 days, and it results in 4 identical sperm. It goes through meiosis 1 and meiosis 2, from our spermatogonia to 4 mature sperm. Oogenesis, on the other hand, actually begins during prenatal times, so before the baby's even born, but it does not complete until fertilization. Several million oocytes are present at birth, but only 400 are ever ovulated. At ovulation, meiosis 1 is complete. So meiosis 1 starts before the baby's born, but it's not complete until fertilization. Completion of meiosis 2 does not occur until fertilization, Fertilization takes place in the fallopian tubes. Once the sperm enters the egg, meiosis 2 completes. And then, so here we have a video on meiosis. Get this to work.
Many organisms pass their genes to their offspring through sexual reproduction. This begins when two gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Many organisms pass their genes to their offspring through sexual reproduction. This begins when two gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have 2 copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have 1 copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are 2 cell division events during meiosis. The first division, meiosis 1, results in 2 unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, meiosis 2, results in 4 unique haploid cells that only have 1 copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with prophase 1. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis 1. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called a recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no 2 siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the two centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are telophase 1 and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis 2. Meiosis 2 is similar to mitosis. The first stage of meiosis 2 is prophase 2. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase 2 and prophase 1 is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase 2, there is no synapsis of homologous chromosomes or crossing over. In metaphase 2, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis 2 began with 2 cells, and each of those cells were split into 2 cells, we now have 4 unique haploid cells at the end of meiosis. These cells are gametes. 2 gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Next, we're going to move on to genes. Genes encode for proteins. The process of using the DNA code as a recipe is known as transcription. So that's opening up our piece of DNA and making a copy of RNA. The DNA unwinds and RNA is made. In RNA, cytosine, adenine, guanine bases are used, but instead of thymine, uracil is used instead. So here there are a number of enzymes called RNA polymerase. It's involved in, first we open up the DNA and then we use RNA polymerase to create a strand using the DNA template. The initiation of transcription is that a promoter, a specific protein known as a transcription factor, begins this process. The DNA strand is 3' to 5' and the RNA transcript goes in the opposite direction. When adenine is present, DNA puts uracil. Initiation of transcription. The promoter is the region upstream that begins the initiation. So it tells us where we're supposed to go. The Tata box is a conserved region of adenine and thymine that tells the RNA polymerase where to attach to. It's about 50 bases upstream from the initiation of the transcription factor. There's also, and further up is the cat box. Once RNA is made, it can actually be spliced, so it makes the pre-RNA and then it splices out the introns. But it actually can do this in different ways. So sometimes, in most genes, it just splices 1, 2, 3, 4, but especially in the immune system, it can splice like 1, 2, 3, or 1, 3, 4. It allows increased number of protein products from a single gene. And this is known as alternate splicing. So splicing out the introns in different ways. A third of genes have this alternate splicing. And again, the immune genes are one of the biggest ones. Another is pseudogenes. There are gene families that the DNA sequence closely resembles a known gene but is non-functional. And this is probably just through the history of, of creating new genes, that some are no longer functional. We actually have about 99% similarity in our genes to that of all animals. Processing, after the transcript is produced, it's then processed. And the way it's processed is there's a cap to make it stable on the 5' end and a poly-A tail on the 3' end. This all occurs in the nucleus, and this allows it to be stable enough to leave the nucleus and go out to the cytoplasm. So here we have gene expression. We make our copy, we stabilize it, we splice out the introns, and now it can go out into the cytoplasm for this transcript to use as a template to make protein. Translation is once we have that completed RNA that's able to leave the nucleus and go out to the cytoplasm, it can now attach to the ribosomes and be used to have the proteins come in and create a protein. So tRNAs are bases that have an amino acid on one side and a specific group of base pairs on the other. And there's 20 different amino acids, but there are 64 different codons for these 20 amino acids. Many of these have more than one of these triple codons, you'll see. For instance, here leucine actually has 4, actually 6. Here's serine, whereas some of these only have a couple. Glutaric acid. But the most important is every single DNA code starts with AUG, or methionine. Every single, this is the stop codon for every protein, and the other is the stop codons, AUU, AUG, and UGA. When that is seen, the protein is then released. So in translation, it always, as I said, starts with AUG, or the code for methionine. The bonding between the codon and the mRNA with the anticodon tRNA brings in the correct amino acid, and when a stop codon is seen, the protein is released from this ribosomal unit. So we have our DNA, here's our cat box, see our tata box, we start a transcript, we have a reading frame, and then we make a copy of the RNA. We then transcribe it to DNA, to a protein, excuse me. And in the protein, it's just basically a number of proteins, linked together, amino acids linked together to create a protein. After that, there's post-translational processing that sometimes there are a number of turns and that the protein needs to make, and sometimes, for instance, in this case of hemoglobin, you need to put like an iron in here. So if there's a mutation that causes the secondary folding not to work right, the iron may not fit together, or the two hemoglobin chains might not fit together. So these are the problems with that. Pseudogenes, these are genes that, like I said, closely resemble but are no longer used. Anything that prevents a gene from being either transcribed or translated will result in non-expression of the gene. Changes in the gene can result in abnormal protein or lack of protein. So study question. RNA is processed to its final structure. Where is the poly-A tail placed? On the 5' end of the transcript? In the intronic region? The 3' end? Both 5' and 3'? Or not all RNA transcripts are given a poly-A tail? And the correct answer is C. Which particular trait is inherited from a parent to offspring? Which is known as Mendel's law, second law. And this concludes this module.